The quantum smell theory suggests that our noses might work like tiny spectrometers, detecting how molecules vibrate rather than just their shapes. Traditionally, it was thought that smells depend on molecules fitting into receptors, like a key fitting into a lock. But this doesn't explain why molecules with the same shape can smell different. For instance, hydrogen sulfide smells like rotten eggs, while its heavier twin, deuterium sulfide, smells noticeably different, even though they look almost identical. Similarly, carvone molecules have two mirror image forms. One smells like mint and the other like caraway. This theory proposes that receptors listen to the molecular vibrations, like recognizing a song by its melody rather than the instrument. It's a bit like tasting chocolate and recognizing its flavor not just by its shape, but also by the unique buzz of its molecules. Traditionally, it was thought that smells depend on molecules fitting into receptors, like a key fitting into a lock. But this doesn't explain why molecules with the same shape can smell different. For instance, hydrogen sulfide smells like rotten eggs, while its heavier twin, deuterium sulfide, smells noticeably different, even though they look almost identical. Similarly, carvone molecules have two mirror image forms. One smells like mint and the other like caraway. This theory proposes that receptors listen to the molecular vibrations, like recognizing a song by its melody rather than the instrument. It's a bit like tasting chocolate and recognizing its flavor, not just by its shape, but also by the unique buzz of its molecules.
Introduction to the quantum smell theory. How do we perceive odors? The sense of smell involves olfactory receptors located in the nasal epithelium, which detect volatile molecules. The classical lock and key model posits that molecules fit into receptor binding sites based on their shape, similar to a key fitting into a lock. However, this model struggles to explain phenomena such as enantioselectivity and isotopic discrimination. The quantum smell theory suggests that olfactory receptors function as spectrometers, detecting the vibrational energy levels of odorant molecules, much like how infrared spectroscopy works. Example: isotopomers such as hydrogen sulfide H2S and deuterium sulfide D2S smell different even though their shapes are virtually identical. This suggests the receptors detect molecular vibrations, not just structure. Analogy: imagine recognizing a friend not by their physical appearance, shape, but by the unique tone of their voice vibration. Similarly, the olfactory system might distinguish molecules based on their vibrational voices. Quantum tunneling in odor detection, a key mechanism. Quantum tunneling occurs when a particle, such as an electron, passes through a potential energy barrier without having the classical energy required to overcome it. In olfactory receptors, quantum tunneling could facilitate electron transfer only when the odorant molecule's vibrational energy matches a specific frequency. This match enables the electron to overcome the energy barrier and initiate a neural signal, encoding the perception of smell. Example: in the G-protein coupled receptors, GPCRs, involved in olfaction, electron transfer might occur only if the vibrational frequency of the odorant molecule matches the receptor's quantum resonance condition. Analogy: think of a radio receiver tuning into a specific station. The electron tunes in to the molecular vibration, allowing it to broadcast the smell signal to the brain. Vibrational spectroscopy and olfaction: how smell feels molecular vibrations. Every chemical bond vibrates at characteristic frequencies depending on the masses of the bonded atoms and the bond's stiffness. These vibrations can be measured using techniques like infrared spectroscopy. The quantum smell theory suggests that olfactory receptors detect these vibrational modes directly. For this, the receptors must have sensitivity to infrared range frequencies, which correspond to bond vibrations. Example: sulfur-containing compounds like methanethiol and ethanethiol have distinct vibrational frequencies due to the sulfur-hydrogen bond, which correlates with strong, unpleasant smells. Analogy: picture a musical instrument producing a distinct note based on its structure and material. Similarly, each molecule plays a unique vibrational note detected by the olfactory receptors. Isotopes are atoms of the same element with different numbers of neutrons, leading to differences in mass, but not chemical behavior. The vibrational frequencies of molecular bonds depend on atomic masses, so isotopomers have slightly different vibrational spectra. Humans and animals can distinguish between isotopomers, a phenomenon that cannot be explained by the lock and key model. Research on acetophenone showed that fruit flies could differentiate between hydrogen and deuterium-containing versions of the molecule, despite identical shapes. It's like playing two identical guitars, one strung with thicker strings. The thicker strings produce a deeper sound, and trained listeners can tell the difference. Critics argue that the olfactory receptors are embedded in a highly thermal and noisy biological environment, making it unlikely for them to detect quantum vibrations with sufficient precision. Thermal fluctuations in the body, approximately 310 K, might obscure the fine vibrational spectra of molecules. Additionally, quantum tunneling might not occur on the timescales required for odor detection. Thermal noise in biological systems is often orders of magnitude higher than the energy differences between vibrational modes, which could disrupt the detection of these subtle signals. Imagine trying to detect a faint vibration in the ground caused by footsteps while standing next to a loud construction site. The noise makes it challenging to isolate the signal. While the lock and key model explains broad molecular recognition, it falls short in addressing why structurally similar molecules can smell vastly different. Quantum vibration theory provides a mechanism where quantum energy states of the molecule interact with receptor proteins, leading to a more nuanced recognition system. The combined approach of both models might offer the best explanation. Compounds like carvone exist in two enantiomeric forms, smelling like spearmint and caraway, respectively. While the lock and key model explains receptor binding, the vibrational theory could account for subtle differences in their perceived odors. The lock and key model is like using a physical key to open a door, while the vibrational model adds an electronic code that fine-tunes the access. Experimental validation includes isotopic substitution studies, behavioral assays, and computational models. Behavioral studies have demonstrated that animals and humans can distinguish isotopomers. Additionally, density functional theory, DFT, calculations have been used to simulate how molecular vibrations interact with receptors. Example: in a landmark study, Luca Turin and colleagues trained fruit flies to avoid one isotopomer of acetophenone, showing a statistically significant preference for one over the other. Analogy: training dogs to detect different scents can be likened to these experiments, but instead of macroscopic smells, the flies smell quantum-level differences. Scientific explanation: artificial olfactory systems could leverage vibrational spectroscopy to detect and categorize odors more precisely. These systems might use nanoscale sensors to mimic quantum tunneling and vibrational detection, improving scent identification for applications in food quality control, medical diagnostics, and the fragrance industry. Example: the perfume industry could design molecules with specific vibrational signatures to produce desired scents, potentially leading to revolutionary fragrances. Analogy: instead of relying on trial and error to mix perfumes, a quantum olfactory scanner could provide a precise recipe based on vibrational spectra. Scientific explanation: the quantum smell theory highlights the emerging field of quantum biology, which explores quantum effects in living organisms. Processes like photosynthesis, magnetoreception in birds, and enzyme catalysis have been shown to involve quantum coherence, tunneling, or entanglement. If validated, the theory would extend the reach of quantum mechanics into sensory biology, challenging our understanding of biophysics. Example: quantum coherence has been observed in the Fenner-Matthews-Olson complex in photosynthetic bacteria, where energy transfer is highly efficient due to quantum effects. Analogy. Quantum biology is like discovering that a hidden layer of quantum magic operates behind the scenes of biological systems, much like uncovering hidden gears in a well-functioning clock.